É um prazer estar aqui mais uma vez no nosso webinário. E hoje nós teremos um webinário internacional, em que também estamos fazendo em conjunto com um excelente simpósio que nós tivemos aqui, coordenado por nossas colegas a doutora Alda Maria da Cruz e a doutora Maria Fátima Fantini, em que ficamos 2 dias conversando e discutindo com excelentes palestras sobre os parasitas intestinais. Espero que possamos ter novos simpósios como esse no futuro e que a Sociedade Brasileira de Parasitologia estará sempre colaborando e divulgando esses tipos de evento. Então, eu só gostaria de lembrar que existem vários eventos para acontecer agora até o final do ano. Eu só vou falar rapidamente alguns. Essa semana, na semana que vem, começa um curso de vacinologia, que o coordenador é o professor Alexandre dos Reis, lá de Ouro Preto, em que vai se discutir as vacinas, desenvolvimento de vacina para leishmaniose e outras doenças protozoárias negligenciadas. No dia 7 de outubro, nós teremos uma homenagem aos 50 anos da revista de Patologia Tropical, que é a revista que a sociedade também colabora. No dia 25 de outubro, nós teremos uma palestra do Pré-Congresso Internacional de Xistossoma, e por acaso eu vou dar essa palestra junto com com a doutora Lisette Lichhout. Nós teremos o congresso do COIRMAN, né, da Medicina Tropical, TropMed, no dia 26/10, e teremos também o encontro de toxoplasmose, esse ano acho que é internacional, né, Alda? E vai ser no dia 23/11. E essas informações estarão todas na página da nossa Sociedade de Parasitologia. Então, mais uma vez, eu gostaria então de agradecer aqui a participação do doutor Alexandre Januário da Silva. Sempre muito bom a gente se encontrar. O Alexandre foi aluno meu de mestrado, doutorado, e colaboramos muito tempo em pesquisa. Colaboramos aqui no Brasil, depois nos Estados Unidos, tivemos a oportunidade de trabalhar com os bichinhos lá, que não são mais parasitos, né, Alex? Viraram fungos, que eram com microsporídeo, e na época ele fez um trabalho de tese de doutorado excelente, foi orientado pelo Dr. Norman Pianazek e por mim, e depois ele permaneceu durante vários anos no Center for Disease Control, desde 92 até, não lembro direito, ele pode falar depois, e saiu, mais ou menos, 2015, mais ou menos, 2000 foi, né? Saiu e foi para o Food Drug Administration em Washington. Hoje ele está chefiando lá o laboratório de parasitas, vamos dizer, acho que é mais voltado à biologia molecular de parasitas, fazendo estudos, então, principalmente em Cyclospora. Então, hoje ele se ofereceu aqui, e a sociedade, ele é sócio da sociedade, sempre foi sócio da Sociedade de Parasitologia, mesmo estando nos Estados Unidos, e se ofereceu hoje aqui para apresentar e coordenar esse seminário, no qual nós convidamos 2 pessoas, o Maurício Durigan, que tem no seu currículo, o pai dele, que é um pesquisador muito conhecido no Brasil, que todos conhecem. Desculpe, mas eu acho que o Maurício seguiu esse caminho, trabalhou com a doutora Maura, lá em Campinas, e, se não me engano, e depois foi para os Estados Unidos, está lá agora nos Estados Unidos. E a doutora Ness, que eu pouco conheço e vou ter o prazer de conhecer hoje, foi um prazer e obrigado. Estamos aqui também com a presença da doutora Alda Maria da Cruz e a doutora Maria Fantinati, que foram as coordenadoras do evento. Eu gostaria de passar a palavra rapidamente, Alexandre, antes de passar para você, se a Alda e a Maria tiverem alguma consideração a fazer, e tenha um bom seminário, então, a todos.
Boa tarde a todas e todos. Gostaria de agradecer ao professor Peralta, né, pela parceria, né, e pela oportunidade que nós tivemos de de trabalhar juntos e a ótima coincidência do nosso evento acontecer justamente nessa semana em que haveria esse webinário sobre Cyclospora. Então, acho que isso marca, né, mais uma vez a nossa integração e como a Sociedade Brasileira de Parasitologia tem sido importante, né, para alavancar vários desses eventos. Então, em nome aqui da organização, eu queria agradecer ao Mauro e à sociedade por todo o apoio, né, e por essa parceria. Muito obrigada.
Eu também gostaria de agradecer, primeiro, a todos os participantes do nosso evento, que está se encerrando oficialmente agora, depois dessa apresentação dessa atividade, e agradecer muito ao Dr. Serão, em nome da Sociedade Brasileira de Parasitologia, por todo o apoio ao longo do nosso evento, a participação ativa nas nossas atividades. Isso demonstra realmente que nós estamos ancorados e estamos tendo esse suporte dentro das pesquisas parasitosas intestinais da sociedade. E eu acho que essa atividade aqui de hoje vai encerrar com chave de ouro, porque os palestrantes realmente vão trazer bastante informação para a gente, bastante novidade, e vai enriquecer muito o nosso conhecimento sobre esses parasitas. Muito obrigada.
Okay, thank you, thank you. I'm sorry to speak in Portuguese, but now I go to change to Alexandre. We're going to present the two speakers in English, and they're going to speak in English also. Alex, up to you.
Okay, well, again, it's a great pleasure, to be part of one of the events from the Brazilian Society of Parasitology. Of course, I am a big fan of the society, and always very glad to be involved with you guys. And actually, I miss a lot because, I'm going to give you like a couple of aspects of the work that is done at the FDA, but I miss a lot, the diversity that the society, provides, and I'll explain why. So at the FDA, the three of us, Nisha Sinha, Dr. Nisha Sinha, Dr. Mauricio Dorigan, and myself, we have been working at the FDA for, on parasitology for a few years. The major, why the US FDA, the US Food and Drug Administration, is concerned, with parasites, right? Because of outbreaks, because of the major, let's say, impact that some parasites have on food safety in the United States. You guys are not going to be surprised by everything that you're going to be hearing today is about Cyclospora. When we say we work in parasitology at the U.S. Food and Drug Administration, that's a synonym for Cyclospora because that's a major parasitic problem here in the U.S. So the Cyclospora, was discovered in there were several, papers showing, that there was a parasite, that there was something that was causing disease in humans. And then, between 1993 and 1994, a group in the University of Arizona, which includes Dr. Inés Ortega, finally came up with a way to characterize and then, made it, created a taxon for that, entity. So Cyclospora cayetanensis, and the name cayetanensis is because most of the studies were done in the Universidad Peruana Cayetano Heredia. So that's why the name cayetanensis came from. And I've heard a lot of people, complaining about because, in America, that's a difficult name to pronounce. But Cyclospora infection, spreads mostly through water, food, and not from person to person. And this is because, the organism is shed by sick individuals, and it takes some time in the environment to become mature, to become infectious. the prevalence of cyclosporisis, and it causes a disease, called cyclosporisis, which is an intestinal, infection, and which can be very severe, in certain individuals. It can last for a long time too. It can last for 45 days in individuals who cannot be treated well, cannot be treated with the main drugs that exist, as a first line of treatment, which is basically a sulfur drugs, right? So the prevalence of cyclosporas increased quite a bit in countries like US and Canada. We have surveillance for that. And according to the CDC, the Centers for Disease Control and Prevention, there was probably like 6,000 domestically acquired cases in the past 3 years, right? And they're all seasonal. They happen normally, between May and August. But we believe that this number is actually higher than that. We believe that, the prevalence of cyclosporas is much higher than that during the times that the disease occur, right? So those outbreaks, I think we experienced the very first outbreak of cyclosporas in 1996, some time ago, 1995, 1996. But recently, those outbreaks have become a major problem, affecting like 2,000 and plus individuals, in 3 or 4 months, right? And more recently, these outbreaks have been linked to produce that is produced by major, let's say, companies in the US. So it's not only a public health issue, it's also a regulatory issue. And that's why FDA, it's working on that. And, like Mauro said, Dr. Peralta said, we go way back, and, dating from the times that I work at CDC, and at CDC, we also had, major programs, on cyclosporas, but the FDA, program is something new. And one of the things I wanted to highlight about the recent, efforts is that the FDA has released something that it's called a Cyclospora Prevention Response and Research Plan, and you might be able to find this plan on the FDA website. If you go to the FDA website and then do a search, actually, if you do a search on Google, you're going to find that. If you do a search on the Cyclospora Prevention Response and Research Action Plan from the FDA, you're going to find it. The research, the results and the topics that you're going to hear today are completely aligned with this research plan that was launched by the agency. So now first we'll have, Dr. Mauricio Durigan, who has been working in environmental sciences, and he's going to, he's going to cover, the environmental detection of Cyclospora. Dr. Durigan got his, he started his studies in Brazil, and moved to the US, in few years, I believe it was like, between 2016 or 2000, I think it was in 2016, more or less. Dr. Nisha Sinha, who has been working for FDA, longer than I, when I joined the FDA, she was already, working on several aspects. She's going to cover, the genomics of Cyclospora, right? And Dr. Sinha, initiated her studies in Turkey and, moved to the US, and had different, let's say, positions in different institutes, in very, reputable institutes in the US, and have been working for the FDA for, approximately, 15 or 16 years, if I'm not mistaken. So both of them are going to provide you guys with, a very nice coverage of how the FDA is tackling Cyclospora from, the environmental point all the way to the genomics, all the way to the analysis of the genes. And, Nisha's, I'm pretty sure that she's going to talk a lot about the difficulties of how to do this, because this is not a very easy parasite, to work with. And with that, I will, let the speakers, take the stage. And, we're going to start with Dr. Durigan, and then, followed by Dr. Sinar. And at the very end of the two talks, we're going to open for discussions and questions. Again, thanks so much for your participation, and it's a great pleasure, to be here with you guys once more.
Good afternoon, everyone. Thanks, Dr. Peralta, Dr. Alda, Dr. Maria, and Dr. Alexandre for the invitation to be here. It's a real pleasure for me, since I obtained my PhD from the University of Campinas, Unicamp in Brazil. So let me share my screen, and I can start my presentation here. Okay. So the title of my presentation is Cyclospora Keratinosis, Method Development for Detection in Water, Regulatory Testing and Implementation. I work for the FDA CFSN. And first of all, just as a brief introduction, Cyclospora keratinosis is a coccidium parasite, and it causes cyclosporisis. As you can see here on the right of the screen, is the life cycle of Cyclospora. It's the only species of the genus known to infect humans, and human may become infected when you consume fresh produce or water contaminated by the sporulated oocysts of the parasite. As Alex da Silva mentioned, when the human host sheds the oocysts, it's unsporulated. So it needs a few days, something between 7, 10 to 14 days, for these oocysts to really sporulate and become infective. That's why we believe that direct person-to-person transmission is unlikely. When you look to the global impact of the disease of cyclosporisis, we can really see that it's considered a global public health issue, because we can find cyclospora in so many different countries, and you have outbreaks in different countries as well. But talking about the US, since the mission of the FDA is to attend the US, in the United States, foodborne outbreaks of cyclosporisis have been reported annually, and there has been a continuous increase of reported cases since 2013. If you look to the right, we can see here a paper of the outbreak, of the 2013 outbreak, and it was a multi-state outbreak with thousands of cases. And when we look to a specific year, to a specific outbreak, even in one outbreak, we can find multiple subclusters in different states. This is the list of outbreaks, of foodborne outbreaks of cyclosporisis since 2000. And this list ends in 2015, so it's really outdated. In this list, we have 32 different outbreaks of cyclosporisis, but only in 14, so less than 50%, we could identify, the researchers could identify the food vehicle. So the source was not identified in most of the outbreaks. In the FDA, we should develop new methods. We need to develop new methods to identify the parasite. And this is the slide for the method development, validation, and implementation program. And everything starts with the method development. But then we need to validate this method. First, we validated the method in our laboratory, but then we should involve other laboratories to help us in the multi-laboratory validation. That's when the method gets really validated for the FDA. Here is one example, one important example for us. When we validate a method, we publish this method in a book. It's the BAM, it's a Bacteriological Analytical Manual. In this example here, it's the chapter 19B, is the molecular detection of Cyclospora keratinosis in fresh produce, and it was published in 2017. It was initially published for detection of Cyclospora in cilantro and raspberries, but then the method was extended to other matrices like carrots, basil, blackberry, cabbage, romaine lettuce, and parsley. So in 2018, we have more outbreaks. We have actually at least 2 main outbreaks. One was in a very famous fast food, and the second one was in vegetable trays that contain broccoli, cauliflower, carrots, and dill dip. And the FDA stated that by using a novel method for detection of Cyclospora in produce, the FDA was able to detect the parasite's DNA in one of the packages of the bagged salad mix sold by the restaurant. So we can conclude that advances in laboratory detection of C. keratinosis were of crucial importance, because only with these methods we were able to have the results to find the parasite in the fresh produce. And this is the first slide in my presentation in which you can see the importance of the validation of methods for regulatory testing and outbreak investigation. Then we move to the water. And we have many different sources of water. We have drinking water, surface water, recreational water, agricultural water, and many others. And the pathogen can be viable in all these sources. That's why they are the pathogen in this source of water. They are very important to us because they can cause outbreaks. They have a huge impact on public health because you can be infected in one of these sources. For the FDA, the first goal was to develop a method for detection in agricultural water. And the potential use of contaminated water for agricultural purposes poses a serious threat for millions of people, because it's possible that this water plays a significant role contaminating crops during the irrigation process. So water testing may be necessary, may be performed during farm inspections to identify potential source of infection and provide support for outbreak investigations. But you have many challenges when dealing with environmental microorganisms. The first one would be the dispersion in the environment. It's very different when you compare one clinical sample from like stool samples to environmental sample. And in a clinical sample, sometimes we can find millions of oocysts. But in an environmental sample, when you look to the microscope in filtration of 1 liter, 10 liters of water, it's hard to find even one oocyst. The second challenge will be inhibitors, because it damages a lot the molecular detection. We have also a lot of turbidity change due to the weather. So we go to the river one day and the turbidity is like 3.5 NTUs, but we go the following day and the turbidity changes to 25 NTUs. And this can really impact the method. There is also an uncertainty regarding where to sample. And the most important, one of the most important is the time delay to identify source and to collect the samples. That's because if the Cyclospora contaminates the produce today, it's gonna take a few weeks for this fresh produce to be at the grocery store. And then somebody eats that fresh produce, gets sick, takes more few days or weeks to have the symptoms, and then you need to identify the common source to go back to that produce, or maybe to that river or the well that irrigated that crop to collect that water. Sometimes it can take more than a month. When you talk about filtration or concentration systems, in the history of parasitology, we have many different possibilities. The ICR method, the membrane filtration method, even the flocculation in calcium carbonate. We have the EPA 1623 method for detection of Cryptosporidium and Giardia in drinking water. We have also the dead-end ultrafiltration, the DEUF. And talking about the filtration systems, doesn't matter which system, but in all of them, we are trying to concentrate the parasite in a robust filtration. And this parasite is trapped in the filter. And after that, we need to find a way to recover this parasite outside of this filter. We need to use a backflush solution, usually, to recover this parasite from the filter in a lower volume. And with this, we start our method development. This project in the FDA started with an interagency agreement between the FDA and the CDC to compare different methods. They were comparing the EPA 1623, the dead-end ultrafiltration, and also a CFC method. And the results showed equivalent detection sensitivity between the DUF and the 1623. But the dead-end ultrafiltration was less prone to clogging, because when you filter water, depending on the turbidity, the filter can clog. And the DUF was also more practical to use in the field. So this was the method of choice for the FDA. And then we started with the development of standards for detection of C. keratinosis in water. We need to develop a framework to evaluate the performance of the method, to say that it was really a good method for detection of Cyclospora. Here in this picture, in this slide, we can see the filter, which is the most important part of the protocol. We can see in the center the bit beater, that would help us in the DNA extraction. We can see the pump, which pumps the water through the filter. And this is the setup of the system in the field. So this is the real setup. We need to get close to the body of water, and you need to adjust the filter to be close to the pump. We need also a battery in the field to provide energy to the pump. And then the good part of this method is that when you do the filtration, you can simply stop, close the filter, and ship it to the laboratory. The analytical part you do in the laboratory, but not in the field, right? This is the setup of the backflush that we perform in the laboratory. We need to use a backflush solution, the peristaltic pump, to pump the solution to the filter in the opposite direction. And then we recover the backflush, which contains the oocysts of the parasite. And then we proceed to the DNA extraction. We use a bit beater for the DNA extraction. And for the water method, we needed to include a purification kit because of the inhibitors that I mentioned before. These are the results of our single laboratory validation, and you have two main important results on the screen. On the left, we show the importance of the purification kit that we use it to this method. The molecular detection, I forgot to mention, the molecular detection in this method is based on real-time PCR. So we are talking about CT values, and the lower the better, right? And before the purification, the CT values were very high. After the purification, they were very low, and that's what we want. They really removed the PCR inhibitors. And on the right, we can see there is the summary of the results of the filtration of irrigation water seeded with different levels of Cyclospora. We seeded the water with 200 oocysts, 100 oocysts. We needed to know how powerful our method was. And the good part is that our method was good to detect 6 oocysts of Cyclospora keratinosis in 10 liters of water. And the results were approved by the multi-validation committee for the FDA. And then, as a final part of this first validation, we tried to use our method in real field samples. So we went to a river into water canals, here close to Washington, DC, and we collected some water samples. And when we went back to the lab, we tested our dead-end ultrafiltration in these water samples, and we were able to find Cyclospora keratinosis in these samples as well. So the method was good also for real field samples. So these results were published, and the journal is here. And if you want to take a picture, we can see the QR code here on the screen. So we finished the single laboratory validation. Then we were ready to proceed with our biggest challenge at that time, a multi-laboratory validation. That was necessary to really validate the method and to publish the method in the BAM. We need to involve other laboratories. We started with 11 labs. They were all trained for the method, all equipped, part of all labs from different parts of the United States, even private labs. And this is the workflow of the validation study. Part of the study was performed in the originating lab, which was the ORSA and the Moffitt Center in Chicago. And we were responsible for sampling the water, for seeding the water with oocysts, and then performing the filtration with the dead-end ultrafiltration technique. Then we shipped all the filters to the collaborating labs, and they were responsible for the backflush, concentration, DNA extraction, and molecular detection based on real-time PCR. And they needed to reproduce our results for the method to be validated. And just to have one idea of the amount of water that we need to collect and to perform the filtration in Chicago, we consider that for each filter, we needed to filter 10 liters of water. And we had at that time 8 laboratories involved, plus 2 for each round, okay? And so it was 10. And we had 3 different seeding levels of Cyclospora, and it was 8 filters per round. So per round, we were filtering 800 liters to ship to the labs, but we performed the study in 4 rounds. So we needed to perform the filtration and seeding with oocysts in more than 3,000 liters of agricultural water. Here we have some pictures of the sampling and the big carboys that we needed to use to collect the water. Here in the lab, we were doing the filtration part in the laboratory at this time because we were doing the seeding experiments there. And then we started packaging all the filters to be able to ship these filters to our collaborating laboratories. In the end, the FDA completed the multi-lab validation of the Cyclospora water method, and the method was approved for use in 2019. The detection limit, as I mentioned, is 6 oocysts, and the method was published in the BAM as a BAM Chapter 19C. So the food method is the BAM Chapter 19B, and the water method, the detection of Cyclospora in water, is the BAM Chapter 19C. And just to provide a final example, we published this method in 2019. And that's the second opportunity here to mention the importance of the validation of methods for regulatory testing and outbreak investigation. In 2020, last year, we had, again, multiple outbreaks of Cyclosporisis, and they were found to be linked to different produce items. Infections were linked to a bagged salad mix containing carrots, red cabbage, and iceberg lettuce. And the FDA started the investigation, trying to find which of these source could be the one, and they decided to collect water because the red cabbage was linked to some farms in Florida. And they decided to collect water in some close to these farms in Florida and to see if the water was contaminated or not. So the FDA stated, again, for the first time, a new method developed and validated by the FDA to sample for C. Keratinosis in agricultural water was used in a field investigation. And it was possible to detect the presence of C. keratinosis in the surface water of a canal near a farm suspected of being a source of the red cabbage. So it was another example how important is the validation of methods. Now we have the tools for detection of Cyclospora or in the fresh produce or in the water. The FDA has pioneered different approaches for these outbreak investigations. We have a few pictures here of field applications. Sometimes the weather doesn't help, right? This happened, that was a snowstorm just the night before the sampling in Chicago. We need to collect a lot of water and put all this water in the truck. And we need to go close to collect the water. It's not easy to collect the water far off the water. So we need to get inside sometimes. Here, I already showed this picture before and how we perform the setup very close to the water to do the filtration on the field, right? When we need to investigate the water, we need to perform the filtration in the field. We have also different research projects, the validation implementation of these Cyclospora methods in different laboratories, the extension of the current methods to new matrices. We are developing new generations of detection based on qPCR or conventional PCR. We are trying to find Cyclospora even in surface water and in wastewater treatment plants, and try to understand factors associated to the contamination of produce, and also trying to implement a genotyping tool for Cyclospora. We have many different projects happening right now. So this is the last picture of my presentation. We are trying to implement the method in different labs in the United States. And now we have a big list of a lot of labs that they are now able to reproduce our method. So thank you very much. This is also our parasitology group and our management as well. Thank you very much for the opportunity to be here. And the next speaker will be Dr. Nisha Sinar. Thank you very much.
Hello, can you hear me?
You have to wait a little because you have a different time to the YouTube.
Okay.
You can speak now. That's fine.
All right. I would like to thank the organizer for this invitation to share our research here with you. So my talk is going to be about Cyclospora keratinosis genomics, and particularly focusing on our data and development of methods on genomics and genotyping, especially for food and environmental samples. I am a research biologist in U.S. Food and Drug Administration. First, an FDA disclaimer. The views in this presentation represents my opinion, not necessarily official policy of the Food and Drug Administration. So the first slide, I would like to give you an idea about the outbreaks in the United States. Here in this slide, you see a map, outbreak map of this year, 2021, from CDC. And as you see, it's a multi-state outbreak, and we see this type of outbreaks in general. In the data part of the slide, you'll see the data about the outbreak, case counts, states involved, hospitalizations, and fortunately, we don't see any deaths. And from 2013, year 2013 to year 2021. As Alex mentioned, that these outbreaks, and Mauricio mentioned, we have been seeing these big outbreaks every year, multi-state outbreaks, and the numbers, if you see the numbers are increasing, partly because we have better diagnosis tools, and partly because maybe it's more prevalent now. So this is, again, this is an underestimation in terms of numbers. Most probably, the real numbers are much higher than these. So in this slide, you'll see the microscopic images of Cyclospora keratinosis. It is an apicomplexan coccidian protozoan parasite, and it is spherical, round shape. The size of the oocysts are 7.5 to 10 microns. In the first panel, you see unsporulated oocysts, and this one is the sporulated oocyst with 2 sporocysts inside the oocyst. And this form is a sporulated form, is the infectious form of the organism. And each sporocyst contains 2 sporozoids. And sporozoids are the life forms can invade intestinal epithelial cells, and the life cycle happens inside the cell, and then the mature unsporulated oocysts are released to the environment in human stool, and then sporulation happens, occurs in the environment, as Mauricio mentioned, in 1 to 2 weeks. So a couple of important information about Cyclospora keratinosis. It has a large genome, large meaning much larger than other microscopic organisms like bacteria genomes, 10 times larger than a bacteria genome. It's about 45 megabases. And it's a eukaryotic organism, again, different from bacteria. Because FDA mostly, especially in the food contamination, dealing with bacteria species, also viruses, this is a different type of challenge for FDA. And it's an unculturable organism. To this date, we don't have any culture system to propagate the organism. We don't have any animal model. We don't have any like cell culture system. So basically, we are, all our research is based on the samples we get from sick patients, like sick people. So the stool samples. This is a big limitation. And also there is another major, I would say, like bottleneck for this research, very low contamination levels in food and environmental samples. So we need to find a way to get genomic information out of, from like very few oocysts. So how do we do that? First, a little more about the genome of the organism. Because it's a eukaryotic organism, it has a couple, like several genomes. So this organism has 3 genomes. Nuclear genome about 45 megabases, and 2 organellar genomes. One is mitochondrial genome, which is smaller in size, 6.3 kb concatemer genome. And the other one is apicopilas genome. It's larger, 34.1 kb circular genome. We started this research in FDA around 2013-14. And at that point, we didn't have much genomic information. When I first started the project, I did some like simple database search, and what I found was only about 3 kb worth of genome information about the organism, to my surprise, because at that point, we already had next-gen sequencing, and we had like genomes of several different organisms. My thinking was like, okay, we have this very important organism in terms of like public health, still we don't have the genome. What would be the reason for that? So we'll talk about challenges in this slide. So what is the challenges for C. Keratinosis genomics. We talked about absence of culture method. So this is a huge bottleneck because we are limited with isolates recovered from patients' stool samples. And that's the only source for biological source for our research. And with this, even though we have ways to purify this oocyst from the fecal matter, still purified oocyst contains some human, plant, or bacteria contamination, bacteria DNA as contaminants. So we never have the sample totally purified. And another challenge is a very resistant oocyst wall. Mauricio talked about the beating for DNA extraction. So DNA extraction in itself is another challenge to break out the oocyst and get the DNA out of the oocyst. So here in this slide, I will tell you about the methods we use starting from the clinical stool sample to next-gen sequencing data. So what do we do? First, we purify the oocyst from clinical stool samples. For this, we use sucrose and cesium chloride density gradient centrifugations. And from one single sample, we usually get 10 to the 5 to 10 to the 6 oocysts. Again, depends on the sample. We may get very little or more. And the next step is DNA extraction. Once we have the purified oocysts, as I mentioned, those are never totally purified. We still have some remnants of flora and human DNA. And then we do DNA extraction using Zymo Research fecal DNA mini prep kit with omnilyzed lysis kit. So we developed all these in our laboratory. At least we did some modifications using the methods from literature used for different organisms, and we modified those to our purposes. Again, typical total DNA yield from a single patient sample is about 2 nanogram to 10 nanogram per 100,000 oocysts. So this is a very, very tiny amount of DNA. And the next step is ultrasonic DNA fragmentation. We use Covaris for that to fragment the genomic DNA to 300 to 1,000 base pair. Then we generate genomic libraries for these samples. And these libraries are used for next-gen sequencing. We use Illumina system, Illumina MySeq, and a typical sequence yield is about 2 to 5 gigabytes. By following this workflow, the first achievement we got was complete mitochondrial genome of Cyclospora keratinosis. The reason for that, when we first started, we started accumulating some genome information, but it was mostly mitochondria genome. The reason for that we'll discuss more in the future, but because it's a multi-copy genome. So if you have one nuclear genome, then you have many mitochondrial genomes in that sample. So then you'll get more of that information. So this genome is 6.3 kb concatenated genome. It's not circular, but the mitochondrial genomes tend to become added to as a repeat type of genome. It's an atheric genome. It has 3 protein coding genes and several subunits of small subunits and fragmented ribosomal RNA genes. So this was our first publication, and you can reach to this genome in the link below in this slide. Next achievement was to sequence complete apicoplast genome of the organism. This is a larger genome. It has more challenges. First of all, it is like highly AT-rich genome, and in itself, it is a problem for sequencing in molecular biology, this type of attribute in a genome. It's a circular genome. It's sort of like remnant of a chloroplast genome in structure, but much smaller in this organism. And it has repeats, 5.2 kb inverted repeats shown in red in this slide. Again, we published this paper, and you can reach the genome information from GenBank with the link below. Then with all our research and improving our workflow, finally we were able to sequence a reference genome assembly of Cyclospora keratinosis. The total length of the genome is 44.4 kb, sorry, megabases, and this is not a complete genome. It is still a draft, but it is annotated. So at this point in NCBI, we have 37 genome assemblies similar to this one. This is the best one among those, but all others are in very good quality. So all these are published by the FDA and CDC, and some of them in collaboration between 2 agencies. Again, you can reach to this information in the link below. So what I told you so far, we can achieve, like we can get a whole genome information, apicoplast genome and mitochondrial genome, following the workflow I talked about, if we have 10 to the 5 purified oocysts from a patient sample. So again, to summarize, we purify and extract DNA, make next-gen sequencing libraries, and sequence using next-gen sequencing, in our case, Illumina, Ampisig, and then we do bioinformatics analysis. So this is the first part of the talk. Second part of the talk will be focusing more into genotyping assay development. So for FDA, obviously, getting all genome sequence is a good research goal, and for academia also. But for FDA, the main research goal is to develop genotyping assays or detection assays. So Mauricio talked to you about the work on detection assays. So my part is working on developing genotyping assays. So we started this part of the research as soon as we started getting some mitochondrial sequences. So it's not, I would say it's more like a parallel research. So while we are working on whole genome sequencing, because we want to finish the genome to like the single base and close the genome, but it will take more time and new technologies. At this point, we are more focusing on genotyping, and we recently published our first paper on genotyping, and we are in preparation for submitting the second paper. This part, I will talk to you about that. So what's the challenge here? We know that if we have a stool sample, patient sample, we can sequence all these genomes, no problem right now. But if we have a food sample, a cilantro sample, or water sample, how many oocysts we can get from that sample, and what can we do with that much oocysts? So we know that we can get only a few oocysts. So the thing we can do, we can amplify genomic regions, and the wise thing to do maybe to start with the multi-copy regions like mitochondria and apicoplas we talked about. In this slide, I would like to tell you about the advantages of organelle genomes for molecular tracking. As you know, for eukaryotic organisms get the genomic inheritance from both parents, from all ancestors. But mitochondrial genomes, they pass through, like it is maternal passage, so it is more linear than nuclear passage. And then tracking back is easier, and it's been used for other purposes, this information. And so we talked about maternal inheritance, and also we talked about multi-copy nature of mitochondria and apicoplas genomes. In this slide, I will talk to you about FDA and CDC collaboration on this important research. The FDA and CDC have been collaborating on C. keratinosis genomics research since 2014. After Alex joined FDA, he brought the collaboration to FDA. That was a great start for this collaboration. Both agencies are working on developing the Cyclospora keratinosis genomics genotyping assays, focusing on their particular needs. Ideally, both agencies should use the same genotyping assay because we want to link patient isolates to food and environmental isolates. This ideal assay should work on clinical, food, and environmental samples. We want to have that assay we can use on all those types of samples. So what are the sample types FDA and CDC is dealing with? FDA is dealing with food and environmental samples, multiple sample types, and oocyst load is a very, very small, a few oocysts on each sample. CDC is dealing with more human stool samples and single sample type. Here in this slide, I will give you more data about the multi-copy nature of the organelle genomes. Here, these copy numbers are estimated based on 18S RNA gene, mitochondria, and apicoplas genomes calculated based on HSP70 single copy gene using mean next-gen sequencing read coverage. So here you'll see for one HSP70 gene, which is a nuclear genome located gene, you have 67 copies of mitochondria loci, 38 apicoplast loci, and 18S loci. So that's why traditionally 18S has been used for detection. And here you see that mitochondria and apicoplast might be even more advantageous in terms of copy numbers for developing more sensitive detection assays, also for genotyping assays. So based on these informations, the first approach we took to amplify the complete mitochondrial genome using PCRs. So for this, I designed a PCR assay, basically 4 PCR reactions overlapping and covering the complete mitochondrial genome. And once I get the PCR amplicons, I combine them together and make libraries and sequence using next-gen sequencing. So this is the workflow. So you can apply this method to a food sample. In our case, we apply the method to 25 grams of cilantro sample seeded with 200 oocysts, and also applied it to clinical sample. But in this case, we didn't have to purify the oocysts. We just extracted DNA directly from clinical stool sample. So then we extracted genomic DNA, run the PCRs on our samples for overlapping amplicons, and then pull the 4 amplicons and created libraries, and we did next-gen sequencing. The gray part of the slide is showing data analysis, trimming the sequences, mapping those reads to our reference genomes, and doing multiple alignments, and finding SNPs, single nucleotide polymorphisms, and other changes in the genome. And then we did phylogenetics to create phylogenetic trees. Here in this slide, you'll see the single nucleotide polymorphisms. Also, you'll see other changes we find in mitochondrial genomes, some repeat region, distal repeat region, in the panel B. So this research also published, again, the same paper. What we did at the end, we created a evolutionary tree based on these sequences. These are the samples collected in 2014 and coming from different parts of the United States, showed in different colors, and also our old Nepal samples. We applied this, and also we applied the same method to a food sample seeded with 200 oocysts. So here, what you'll see, the samples coming from the same geographical location, they clustered together. So this suggests that this method has a power, at least based on geographical data. It can cluster, it can give us idea about the differences. So this is published. This was published in 2020. So with this, now we have another method. This time, it is much more sensitive in a way that we can get out of complete mitochondrial genomes from a food sample seeded with 200 oocysts. And also we can apply the same method to clinical stool samples. This time, instead of purifying oocysts, we can directly extract DNA from the stool sample. and get complete mitochondrial genomes to be used for genotyping. While we're doing this research, in 2018 outbreak, there was a food surveillance after the outbreak, and based on that surveillance data, we found out that food contamination levels are at 5 to 10 oocysts per regulatory sample. So this means our 200 oocysts seeded sample data, even though it is really exciting, but it's not going to solve our problem. So we need something even more sensitive than this. Then we decided to use Illumina Ampisig targeted resequencing method. This is a streamlined, scalable amplicon sequencing solution producing high confidence data from low input DNA and RNA samples. We're talking about 1 to 100 nanogram DNA as a starting DNA. Still, it is quite high for our purposes, but we wanted to try it. And it's a multiplexed, highly specific PCR-based workflow. So you can think this as a very sophisticated multiplex PCR. And you can multiplex from 12 to 24,000 amplicons in one assay. So this is what we wanted to use. And for this, the first step is to create a panel. It's called panel, covering complete mitochondrial genome, designing primers to cover the complete genome. And our panel contains 35 amplicons. And after design, we get the libraries, prepare libraries using this panel and sequence as we sequenced before on MySeq and analyze the data. So we tested this method on seeded food samples again. So this time we used different types of foods seeded with different numbers of oocysts. Here it's quite a big table. Hopefully I can walk you through this. So here you see unseeded samples. So we didn't seed these samples with Cyclospora. So we shouldn't find any Cyclospora on these samples. So these are negative controls. We found very few sequences. These might be some laboratory contaminants, but very, very small. And like if you see mean coverage, meaning for each nucleotide, like how many read you have. So here you see it's very, very low. Then we call this not determined. So negative controls work well. And then we have our seeded samples. We started with 5 oocysts, 10 oocysts, and 200 oocysts. Even with the samples seeded with 5 oocysts, we got like thousands, hundreds of thousands sequences from mitochondrial genome. And here, coverage means that like for each nucleotide, how many proofs do you have? How many times you had hit that sequence? And these 3 samples, even though the average we had some coverage, but I set up the coverage. If I find something lower than 35, just to be sure, I call it not determined. So I don't analyze that data. So all others are higher than 35, and most of them are much higher than 35 average coverage. And with this data, we were able to recover 98% or upper, more than 98% of complete mitochondrial genome, even with 5 oocysts. And with 10 oocysts, we were able to recover mitochondria from each and every sample, and same with 200 oocysts. So this data is very, very promising, very sensitive. With this, now we can get complete mitochondrial genomes from a food sample only with 5 oocysts, or a water sample, environmental water sample with 5 oocysts. And again, we extract DNA. We also can use this method as we used the previous one with the clinical samples. We tested that it works. So again, we don't have to purify with this method. And it is much more streamlined than running 4 PCRs. Now we don't have to run any PCRs because it is embedded into the library. So we create the libraries and then sequence the libraries and analyze the data. So with this is my last slide. Take-home messages from this talk is that Cyclospora Keratinosis genomics has its own challenges. Mitochondrial genome information has discriminatory power that could be used for genotyping purposes. Illumina Ampisig targeted sequencing is sensitive enough to sequence near complete mitochondrial genomes from food samples contaminated with 5 to 10 oocysts. Next steps for our research program. For the first part, for getting better genomes, completing and closing the Cyclospora genome, we want to improve the whole genome assemblies. So for that, we would like to use new approaches such as Oxford nanopore sequencing, and we would like to sequence new Cyclospora keratinosis isolates from all over the world to enrich our whole genome databases. For the genotyping method development, we would like to improve our current method by adding more targets. At this point, we added a partial apicoplast genome to our method, and we will test that. And also, we are planning to add CDC's approach, MLST targets to our mitochondria and apicoplast targets and increase, again, genotyping resolution. Also, we would like to use other targeted sequencing solutions, such as hybridization capture-based platforms, and even single-cell sequencing as a new approach for our future research. With that, I would like to thank our team in FDA and also our collaborators in CDC. We have very talented student interns coming working with us, learning from us. We are learning from them too. And also we are in collaboration with Texas State Health Laboratory. They provided the samples, 2014 samples for us. And thank you for your attention.
Thank you, Dr. Sinha.
Thank you. Thank you for
the invitation. We have to wait a little because we have a delay. I would like to say thanks to both of you. I think it was a very nice talking. As I know Cyclospora from Alex in the time that he detected the DNA in the cake, in the strawberry cake, in the middle of strawberry cake.
Wedding cake.
Eh?
It was a wedding cake.
Wedding cake. That was very nice.
Raspberry wedding
it's
cake.
a very nice history about the detection of the Cyclospora, and it's good to know that the progress of this research in that area now. I don't know if you have a question, but I can ask something to comment something to Dr. Durigan. It's about the group. We had a collaboration a long time ago, and Alex know very well Dr. Carmen, and from Spain, and she used to work also in the environment sample, but they use the same technique, basically the same technique that we standardize for Cyclospora. They use for Cryptosporidium and also Legionella, and Acanthameba, in fact, with Legionella. That was very nice. And Regina Peralta, she had a collaboration, and they used to have, for better concentration, after all this filtration, they used to collect the small amount of water and use magnet beads with antibody, in this case, antibody against Cryptosporidium. How do you think about the possibility to have more concentrated material? If you use this bead, you need to have a good antibody, also monoclonal antibody against Cyclospora. Do you have any? I don't know the literature about in Cyclospora. There is something published, or do you have something in this direction?
So we would love to have, but we don't have it right now. We know some collaborators, they were working on it. So we tested before, now they are developing maybe in a better way, more beads. Because yeah, that's true. I used to work with these beads, magnetic beads for Giardia. And it helps a lot, right? Because you have like a very a large sample and you have a lot of debris. So the way we do to separate the organisms like the Giardia or Crypto in the middle of this debris, this changes a lot. If you can purify the sample, separating the debris for the oocysts of Crypto and cysts of Giardia, in this case for Cyclospora, this would help us a lot to have more positive results, right? Sometimes we can have a negative result, but is this a real result? It can be a false negative, just because we cannot find it.
And maybe you can have a more purified DNA also, because you have more, the purification is going to be better.
That's right, yes. Yes, the inhibitors, yes. Yes, it changes everything, because sometimes you go to the PCR and it's like negative, and we put a positive control in this water and it's also negative, so it's the inhibitor, right? True. It would help us a lot if somebody comes with the kit, like for Cyclospora Keratinosis, this definitely would help.
Okay.
And I'm gonna add, if you have such reagents, it will allow maybe, and Nisha can comment on that. It may allow a much better, let's say, yield in terms of DNA for genomics, for genotyping.
Yeah, that's true.
What do you think, Nisha?
Yeah, exactly. So we are doing running methods like centrifugations, and so these are the methods we have. But at the top of these, or even without with bypassing these and using an antibody, would be much faster and less labor intensive, maybe much better if we have that antibody.
But there is some antibody. Do you know, Alex, antibody about somebody else?
Yeah, there are some, but they are not. They haven't made, to the research mainstream yet. Some people are working on it. There is a question, from Dr. Nani Matos on the chat, and the question is it's for the in regards to the method that detects cyclospora in water. She wants to know if the water that was tested were treated water, and I believe what she's asking is if the water in which cyclospora was detected was chlorinated or treated somehow.
Okay, so no, the water had not been treated before. We were trying to mimic the agricultural water that we have here in the United States. So we went to a water source. It was a lake very close in this area. And then we collected this water. We tested before, because in our protocol, actually, if you have chlorine in the water, we need to treat this water in a different way. But no, our water was like just the water that you collect in the environment, and you were bringing this water to the laboratory.
And she also wants to know if the method could actually work on treated and non-treated water, in the same way.
Yes, we believe so. As I mentioned, we have a part of the protocol. If you have chlorine in the water, we have tested this method, this protocol, for a little different turbidities. So yes, we believe that the method works, because we are talking about not only drinking water, we're talking about like a wash water, the water that they use to clean the fresh produce inside the companies, in the facilities. We are talking about well water, the water that they put in reservoirs to irrigate the crops. So our method needs to be good enough for all these different types of water. We started with like the raw agricultural water, and then, as I showed in one of my last slides, that we're extending the method now for these other matrices to be sure that it works well in these different matrices.
So another question from Dr. Nani Matos. She wants to know, what was the biggest challenge in terms of detection of cyclospora in water? Was the collection, the purification, the isolation, DNA extraction, or the standardization of the PCR method?
Yeah, I would say we have more than one, I believe. The molecular detection, we followed the fresh produce method. So we had published before, right? Our team had published the method for detection in produce, and we used the same real-time targeting the 18S. And I would say that for this method, one special challenge was the inhibitors. So we need to include in this method a purification step, because in the beginning, our CT values were very high, and this is not good for real-time detection. We needed to find a way to overcome this challenge. But we have many challenges. Another challenge is, like, as I mentioned in the beginning, the turbidity change. So you are using to a pellet, like 1 ml pellet with the turbidity is 3 NTUs. And then you go to the same place in the following week, and the turbidity is 25, just because it rained the night before. So your pellet will be like 3 times bigger. And you need to process this pellet in the laboratory and to try to find cyclospora on it, but you need to split in different tubes, and in the end to have the same amount of DNA. It's another challenge, in environmental, in the field, environmental samples, everything changed from one day to another. So this is what we try. We try to accommodate all these changes to our protocol, but of course, sometimes you can't. Sometimes we have a lot of inhibition. We dilute the sample 1 to 10. We use another purification kit, but sometimes it's hard, to deal with this difference between one sample and the other.
Because that, I believe, the purification is the most important to have.
Yes, for sure. One of the most important
to
parts.
have a pure and good DNA to work with. For us, that work with stool, Alex know very well about that. We remember in the beginning, Then we published a paper about how to get the DNA on the stools and how to avoid the inhibitors. It's a long time ago. Then it's the same. It's the same for environment sample. Like you said, if you go to the chlorine treated water, you're going to have a problem. You need to treat also this sample. If you go to the natural water, you're going to have problem also. Then is a big problem. I think if you have a clean DNA, Sinar can do a nice and good job like she's doing now. And a question for Dr. Sinar. What do you expect to find when you said in your presentation that you have already good, as you show, mitochondrial sequences, and you did a nice job. And you mentioned that it's good to have also the nuclear studies, nuclear sequence. What do you think that you can find between in which step is going to be better to have a nuclear? What to expect about that?
So my vision is to continue using mitochondria sequences, mitochondrial genome, adding apicoplast reads, apicoplast sequences as much as we can, plus adding nuclear targets. The reason for that, nuclear targets are important. First of all, at this point, CDC has a method. They are mainly using nuclear targets. They are using an MLST method, targeting, they have 8 targets, 6 of them from the nuclear genome, 2 of them from mitochondrial genome. So with that, by adding at least CDC's targets, we can have a common talk between 2 agencies. So if we have the data, if we can, because as I talked before, the challenge is our samples are very, contain very little amount of Cyclospora contamination. So we can get mitochondria because it's multi-copy. We may get apicoplast, I don't know yet, because I didn't test the new panel. But when it comes to nuclear targets, I have some questions if we're going to be able to get nuclear targets. But that's something we need to test. We need to try and see what is the limit for us. And yeah.
The MLST is done in what, in a mitochondrial gene?
2 mitochondrial targets and 6 genomic targets.
Okay.
So there is
Alex.
Yeah, there is a one question for Dr. Julianne Greiner Goulart. And I'll say I'm going to ask this question, and I think I'd like to hear both Mauricio and Nishi commenting on that. And her question is a very interesting one. She's asking, do you think that Brazil also has Cyclosporisis outbreaks? For instance, in the summer, when a lot of people have, let's say, gastroenteritis and intestinal disease. So do you guys think that Cyclospora is causing the problem in Brazil as well?
Anisha, please, you can start.
Okay. So again, without a data, it is hard to tell for sure. It's a possibility because now we are in a global world, and we're getting food from maybe Brazil, and maybe we are exporting food to Brazil, and that's a possibility. Yeah, Brazil may have that problem, but I don't know about their system for surveillance, and so I don't remember hearing any major outbreaks, Cyclospora outbreaks in Brazil.
Yeah, and in my opinion, you just need to apply the methods, right? We need to develop the methods, to bring the methods from different agencies and apply in the country. So I believe if you start using the methods and try to find Cyclospora, you'll find it. Because you have in many countries, right? You have many cases that comes from Peru, and it was the original country that then they published the method and the first cases there. So there is no reason to believe that we don't have that in Brazil. So we just need to go and try to look for it, to find it.
There is a publication about the Cyclospora in Brazil.
Yes.
What's your group? The group of Regina Maura or the other groups? Regina, I think she never found it. I don't know.
Yeah, I remember an old publication. I need to go for it to find it. But the thing is, we need to look more, right?
Yeah, I agree.
Yeah, if you apply the methods, in our group before, we were using for trying to find Giardia and Cryptosporidium in Brazil. And yeah, I was there when we didn't have the beads, the magnetic beads, and then we start buying these beads, and the recovery improved a lot. So that's one example. When you improve the methods, we start seeing things, right? And the same is true for Cyclospora. If you are using the techniques to find in the microscope, to try to find the autofluorescence of the oocysts, that's nice. But you cannot, if you are not concentrating the samples in a small pellet, and try to use a good method for detection, a molecular method mainly, we're not going to find many, So if you apply these nice new molecular methods to the samples and to water, to wastewater, you're going to find it for sure.
Yeah, one thing for sure is that I've heard reports of individuals, who got diagnosed with cyclosporisis, and it was laboratory confirmed. Not only, I think there was a couple of cases from daycare. There were also, back in the days, an outbreak that happened in one of the cities. I don't remember which one, but I think it was in the state of São Paulo. So there are some evidences that Cyclospora circulates in Brazil. The problem, like Nisha said, without a good surveillance system, it's almost impossible to understand what is the real actual public health impact. You need to have that. So there's now another question here from Dr. Jeffrey Shaw. And again, I think it's a good question for both of you. You guys can comment on that. How does the water becomes contaminated with Cyclospora, and are there asymptomatic carriers?
Okay, yeah. I will start.
Go ahead, Nisha.
So as far as we know, this is like human-specific disease, and so the contamination source would be human fecal contamination. That's the major source. And how would it happen in the United States? We have aging infrastructure. There might be some escape from treatment facilities. That might be one reason for maybe the increase of cyclosporiasis. And again, we need more data to find out about this. And we also, there are other ideas about, for example, we don't know how to explain the seasonality of the disease. So is there any other thing, like maybe migratory birds or something like that, carrying the organism, maybe just as a carrier? So all those like unanswered questions we have about like how the water can get contaminated with the oocysts. So that would be my answer for that. And what was the other part of the question?
If there were if asymptomatic carriers exist.
Yeah. So there are reports that about asymptomatic carriers. So yeah, that's also might be a possibility.
And just to complement the answer, yeah, that's one of the most important questions, right? Because we have the problems in the patients first. Then they realized that it was coming from food. And now you are trying to link the water to the fresh produce. And then how it went to the water, right? We had some hypotheses, but now we are at the step of testing of detection methods to test where is the cyclospora. So it can be a human contamination. We found some cyclospora close to wastewater treatment plants, and we know from literature that is possible, right? From these parasites to go through the treatment and to pass these stations. If they found cyclospora in the effluent of wastewater treatment plants. So that's one possibility. But we have many other possibilities, right? The contamination of water is possible in many steps. So I'm pretty sure that that's where we are going in the future. Now we have a method for detection in water. So next steps, where is it coming from?
Question, there is any animal that was found, or birds, or I don't know, Cyclospora, I don't know how's this, is zoonosis or it's not a zoonosis, I don't know what you can say about that.
We don't have concrete evidence. We have a couple of early papers suggesting that there might be dogs or some other animal reservoirs, but none of them were like molecular detection, and then we couldn't find anything more. So at this point, the general conclusion is the organism Keratinosis is human specific. But again, there might be a way, because as you know, oocysts are very sturdy, and animals can carry in their intestinal system and then release it without being an active host for the organism.
Okay.
There is a commentary from Dr. Sidney Silva, and he says the work that they were doing in one of the institutes that sees HIV positive patients were not able to detect Cyclospora in that setting. But he also said that the techniques that were used were the staining techniques, the modified quinine acid-fast technique. So he's saying that maybe that study didn't yield any positives with Cyclospora because of the low sensitivity of the methodology used. And that
I believe so. That's true.
Yeah, and that goes into what Mauricio was saying, that maybe some of the patients, they shed a small amount of oocysts, which it's not very easy to detect. The other thing, I don't know if you guys are going to make this comment. My background is actually oocysts of Cyclospora that fluoresce. Okay, so if you put the Cyclospora stool sample under in a microscope, in a fluorescence microscope that has UV source of light, that's what you're going to see. You're going to see the Cyclospora, in this bright, all those little circles you see there. That's one, each of them is one oocyst of Cyclospora. So that technique is actually more sensitive than acid fast. So it might actually, be advantageous. And you also use wet mounts. You don't have to do any staining. So basically what you do just like a concentration of the stools and put that on the slide, and then put it on a fluorescent microscope with UV light, and with the right filters, you can see, the parasite, producing this autofluorescence. And that technique is described on the CDC website. You can just easily retrieve from that. So that's one of the, let's say I would advise people if they will continue to use microscopy to try the UV fluorescence microscope on Cyclospora, because it might enhance your sensitivity of detection. And there is another commentary, again, from Dr. Sidney Silva. He said that, maybe one of the reasons, and I agree with him, one of the reasons they couldn't find, Cyclospora in those patients was because a lot of those patients had been treated, for pneumonia caused by Pneumocystis jirovecii. And that was a prophylactic treatment, which I believe used Bactrim. And as you know, Cyclospora is extremely sensitive. I mean, you can treat Cyclospora very well with sulfametazole, which is Bactrim, trimethoprim sulfametazole. And maybe he's correct. One of the reasons why they couldn't find Cyclospora in this group of patients is because they were treated, for pneumocystis, which worked on Cyclospora. That is
a good point.
I agree.
Alda, Maria, any comment?
No, thank you.
No.
I think you have a good closed section for this meeting. And I don't know if Sena or Mauricio wants to say the final words.
I would like to thank again for the invitation. This was a great opportunity for me to discuss the research with you, and great questions, great discussion. Thanks again.
The next time you can visit Brazil, let's go to see if you can come in the next parasitology meeting.
That would be great.
We all love it. Just invite us. We all will come.
That's true. Thanks, Peralta. Thanks, Alex. Thanks, Alda. Thanks, Maria. Thanks for the opportunity. It's very important for us to connect to Brazil, to keep talking about our research. Thanks for everybody that watched it. It's a pleasure. And my supervisor in Brazil was Dr. Regina Maura Franco and Dr. Anete Pereira de Souza. So it's a pleasure to be here talking to everybody today. Thank you.
Okay, thank you for both of you. And I hope you can see. I remember Mauricio when he was, I think, 7 years old or something like that. When he lived in Atlanta. When did you live in Atlanta?
No, I went to Atlanta to meet Alex. It was in 2013, Alex, right? I stayed with him in the lab for like a month in CDC.
Okay.
Yeah.
Okay, everybody, we have a nice session. Just remember, I go to speak in Portuguese now, okay? Lembrando a todos que essas palestras são gravadas, todas elas estão gravadas no YouTube e podem ser assistidas, não só essa, como outras também, antigas, e são vários seminários que trazem bastante uma colaboração muito boa. Na próxima semana, no próximo seminário, que vai ser daqui 2 semanas, o assunto vai ser girofilariose, uma infecção canina, que vai ser dado pela doutora Norma e doutor Bueno, da Fiocruz, da UF. Tá? Eu aguardo, então, todos uma boa noite. E, Alex, muito obrigado. I thank you, Alex, for this nice end of quinta-feira, Thursday. Okay? And have a good night for everybody. Bye-bye.
Bye bye.
Boa noite a
Valeu.
todos.
Tchau, boa noite a
Boa noite a todos.
todos.